Good morning. My name is Rupa Pike. I am the Global Head of Strategic Alliances at Catalent. And as the announcer mentioned, welcome to the Science Slam session, Comparability Studies, Walking the Tightrope. So as all of us know, comparability studies are very critical for ensuring that changes in the manufacturing process or different versions of it still need to maintain quality, safety, and efficacy. And successfully walking the tightrope of comparability studies involves striking a delicate balance between scientific precision, manufacturing process, and regulatory considerations to make sure that the product is reproducible without changing its profile and meeting the regulatory standards. So today, I would like to introduce you to our distinguished panel. They each bring a unique perspective on comparability studies based on their experience. They will each offer a short presentation, and we will follow up with a discussion, and we will have time for audience Q&A after that. So the first presentation will be by Karen McIntosh, who is the Vice President and Head of Regulatory Affairs at Orca Bio. Next, we have Alan Calloway, Director, Global CMC Regulatory Affairs at Johnson & Johnson. And last but not least, we have Mike Mendicino, who is the President, Owner, and Chief Consultant and Advisor at Hybrid Concepts International. We will start with Karen. Thank you.
Thank you very much for that introduction. My name is Karen McIntosh, and I'm the Head of Regulatory Affairs at Orca Bio. Orca Bio is a biotech based out in San Francisco, developing allogeneic T-cell immunotherapies for hematological malignancies. This has been a particularly good week for us as at Orca for two reasons. First, we announced on Monday that FDA accepted our BLA application, and for ORCA-T, our lead product, and then the Nobel Prize in Medicine got announced for the work in regulatory T cells, which is one of the main cell components in ORCA-T. So lots of exciting work ahead for us. But we're not here today to talk about Orca Bio. We're here to discuss comparability studies. I would like to approach these next few minutes from a regulatory strategy perspective. I don't have any fancy slides or anything of the kind. This is a discussion topic, and I would like to dive further into how we can plan for these studies, how can we be smarter, and how do we work with the agencies, and most importantly, how do we not fail? Comparability studies are guided by ICH Q5E, comparability of biotechnological/biological products subject to changes in the manufacturing process. This guidance leaves, unfortunately, a lot to interpretation, and I think I speak for many of us when I say that this can cause both a lot of uncertainty and confusion, both within your company as well as with the agency. ICH Q5E applies to the manufacturing processes changes both during development as well as post-approval, and it outlines a stepwise risk-based approach, just as seen in this figure here. We're starting with analytical testing, followed by non-clinical or clinical studies only if necessary. You can see some of the attributes needed for your analytical testing outlined here. And if this moves over to needing a non-clinical study, this will usually be a PK/PD study or a toxicology study. And if a clinical study is needed, that would be PK/PD, bioequivalence, or a bridging study. This approach may seem very logical, but it's very, very hard to manage in day-to-day life for a company. How do you plan for it? You often get asked the question, okay, we're making this change. Are we going to need a clinical study for this? Well, that's really hard to respond to because we won't know until we've done the analytical testing. Or the other way around, where it's like, yep, we're just going to go ahead with our analytical comparability, everything's fine. Oops, it was not. We're now adding on months, if not years, to go into a non-clinical or clinical study. There we go. The interpretation of ICH-Q5e differs not only between people in different departments, between departments in your company, between companies, but it also differs between the different health agencies that have adopted it. For example, as most of us know, FDA lean a lot more towards a risk-based approach, while, especially for small molecules, while the EMA may demand a lot more extensive analytical and clinical data, especially for biologics and larger products such as cell and gene therapies. Other markets outside of these have their own take on it, but may have even more additional local requirements needed. So we have a difference in standards by region. We have a variation in documentation. Adding on to that, there's also a guidance evolution, and especially with dealing with younger health authorities, it may be that when you're making your plan for your comparability study, the requirements look one way, but when you're actually finished it and you've got that report in your hand, all of a sudden they've changed and you need something completely different. Also, timelines change considerably between different regions. One small example up here between the US and the EU for a comparability, this would be for a larger change, let's say, an addition of a manufacturing site, where for the US, this would be a prior approval supplement in the post-approval stage and be anywhere between 4 months to a 10-month approval timeline if a reinspection is needed. So it's hard, again, to say, yeah, this is how long it's going to take. This is when we can implement our post-change product. The EU, slightly different. For a similar change, it would usually be 60 to 90 days. But again, with the complexity of your product, as well as other factors, this could spill into having other issues in the post-approval world. Planning way ahead for these studies is crucial. Not only do we need to adhere to the local requirements and timelines, but we also need to think about our resource planning at the manufacturing sites. These are heavy resource needing studies. The when and how of this planning, as much as I would love for it to be driven by regulatory timelines, is most never so. It is driven by the business need. We need to implement this in such a time. This, can cause delays. You may not have that time to plan appropriately, to take everything into consideration, and this misalignment can affect clinical supply or other post-approval changes. So how do we fix this? Well, back to planning. To integrate the timing of these changes, to allow time for generation of analytical and functional data, and in your project or program meetings, add this to your swimmer plots. Add this, talk about it, plan ahead, make sure it's part of your budget to ensure that the resource is there, that the time is there. Keep any future markets in mind when you're putting together your protocol initially. Are you going into the EU? Well, maybe you're going to need something different than compared to the US. And yes, you also need to really think about it. Often I see companies, they're like, well, we're just in phase 1. We can get away with doing a much lesser comparability here. Not a problem. It goes through, everything's fine. You then get to BLA, that comparability report sits in your BLA and gets reviewed by your BLA reviewer, and they're gonna go, nope. All of a sudden, you have a whole potential phase 1 clinical study you cannot use. So think ahead, don't do this the short way. Another good idea to do is a predefined comparability protocol template where you can go in, you can vet this with your agency before, say, hey, if we follow this, and clarify the expectations for your analytical and clinical data requirements, and have this discussion over and over and over ad nauseum until everybody knows what needs to be done. This is especially hard in our environment in the cell and gene therapy, because in regular drug development, you have a phase 1, a phase 2, a phase 3. Yes, your pivotal study, you want to have your commercial manufacturing process in place. Great. You often, during phase 2, do all these changes. You're updating, you're automating, you're doing all these changes during your phase 2. We don't have a phase 2. We go from phase 1 to phase 3. So either you really pre-plan and you do it during phase 1, or you're going to have to do it during phase 3, causing all those regulatory complications. So this was me from a regulatory perspective, and I'm looking forward to the rest of the conversation later. Thank you.
So good morning. I'm going to take a moment, I think, to, if you will, just set the stage for you. Really want to look at this from a holistic perspective. So comparability is a point-in-time study, but when you're looking more broadly, that's really what I think drives your strategy for development and commercialization. One of the big items we see, and I think Denise Gavin highlighted this about 2 weeks ago in a public forum, the link between CMC and clinical for CGTs is more important than in probably any other modality. So what you will see, we'll kind of go through some of the concepts, look at some phase-specific items. There we go. And then we'll actually go into some watchouts, if you will. So if you're looking at comparability requirements across the board, so most of us think, you know, if we're starting a phase 1 IND in the US, we have that in picture. If you're thinking to globally to commercialize a product, you're looking at US, EU, you're looking at AP, and right now, just by the last count, there's almost 400 individual ATMP guidelines in this space. So if you can imagine if I put that to a phase 1 developer and said, make sure you meet all of these by commercial, they would say, thank you for coming and have a great day. There's no way to do that. But honestly, what you see are regions are building on comparability requirements. So US and EU are somewhat aligned, different requirements. AP is building, and Visa is behind that in the Latin American space. So just basic requirements off the top of your head. CQAs is always kind of your core item. For a lot of our modalities, especially the newer ones, we are struggling, if you will, to identify formal CQAs, which builds into our potency strategy and biological activity. Implementation of analytical strategy. So ICH, I think, is a huge driver in this space. We're starting to see definitely a footprint there. So the new Q1 stability guidance has an ATMP annex. I work actually on Q6. We're talking about ATMPs, broadening the scope for that. And you also see there's a cell and gene therapy discussion group, which is actually clinical, non-clinical, and CMC. So I would say pay attention to ICH in terms of requirements, but also leveraging prior knowledge, which isn't a new concept. It's been around for about 20 years between Q8 to Q12. So design of your characterization, assay strategies, you're going to need this for comparability. Your release panel is generally not going to be sufficient even in the early phases. And careful identification of that pre and post change starting material, the manufacturing process, and your acceptance criteria. I think that long-term strategy, and it may feel like a stretch early on, when you have a pre-IND meeting with the agency, you should be thinking at least 2 years in advance, because you do not want to file an IND and then 3 months later say, by the way, we have a huge CMC change and we have a new clinical protocol coming. Can we have a type C meeting, type D meeting? You have to start to build at least the strategy in play. So I kind of broke this down into the phases. So what are your requirements? What are the things you should be as kind of a watch out? So one, for early phase, you could have limited material availability due to your scale, patient-derived materials. If you're in that rare disease space, that's pretty difficult in itself. I always find that the early phase is more frequent and iterative changes to your process than once you've scaled up and get close to that pivotal process, if you will. Complex CQA identification, product specifications are always, I think, difficult, especially with any sort of donor material as a starting material. And then analytical method development. So your initial methods are going to be to the point of hopefully platform for some of the impurities, but a lot of your other methods, especially bioactivity and potency, are generally going to be product specific. You're going to need to work on those pretty quickly, at least to have a strategy, because FDA is definitely looking at that from a potency assurance perspective. As you get close to your registrational studies, and as Karen pointed out, kind of skip phase 2. So you could have, a very small pivotal study, 100 patients. Even with that excellent clinical data, and we've seen stunning clinical data out of these products, you don't get out of the CMC requirements. So you really have to work with that clinical team to have that discussion. I think one thing here is really looking at what is going to be the comparability assessments as you get close to pivotal. Making a major CMC change in the middle of pivotal is not unheard of, but it's not something I would recommend to you. And I think it's one of those where if you get to a point of automation and you need to make a decision, you also have to decide, because moving a major change in the commercial space, that's going to take you quite a bit of time, and all of those requirements and review timeframes are lengthened. The communication piece with health authorities, I think, is probably the most important that I could stress to you today. When you go in and you say, this is our comparability protocol, here's our acceptance criteria, and the strategy, you have to remember they're not going to give you, a check mark on your study. It's always going to be a data-driven study, but you can at least get some buy-in on the approach you take. Keep in mind, even though you have a study approved, say, by FDA, when you go broadly to EU, they may ask for additional runs. So if you do an N of 3 comparability, N of 4, N of 5 is not unheard of. So you have to somewhat build in, the materials for that, which are always limited, both stability and analytical retains, but also for the comparability study itself. And then lastly, in the commercial space, unfortunately for a lot of our products, we still do development in the first year or two of that commercial product. And if you can imagine the timelines for that, the regulatory hurdles just continue to go up. So I would stress to you, the more you can kind of be forward-thinking a year or 2 years out from where you are, you're not going to have committed plans, you're probably not going to have a KEM that goes with it. But you need to have kind of an idea of where you're moving, process improvement, scale up, scale out, and site transfers. Because every time you run comparability, there's inherent risk. Analytic variability, manufacturing variability, and then just operator-to-operator variability. So you really kind of want to try to build that in as best you can. As you've heard during the conference, there's a lot of folks, we're all kind of running as fast as we can. Patients are waiting. I'm one of those people who used to review the safety reports that come out, so I can tell you it hits home. But it's one of those, sometimes waiting a month can save you 6 months the next year. You really have to be strategic about the decisions you make and not let a clinical timeline run or a CMC timeline run on a hand. Lastly, one thing I encourage is kind of using Q12. So in the post-approval space, this is relatively new. It's about 5 years old, but hasn't been fully uptake by all health authorities. And this is really looking at that post-approval space. If you're going to do comparability, I suggest using the post-approval change management protocol. It's a way to kind of brief them on what you're doing. Protocol design, get buy-in before you execute. And it may give you the ability to actually downgrade your post-approval submission as well. And then lastly, just some key considerations and opportunities. So if I could get a tattoo on my forehead, the first one would be it. CMC comparability is data-driven, science and risk-based. Protocols are nice until they see data. Doesn't really do much for you. If you have small-scale models, prior knowledge, platforms, all of this builds, even directional data from the early phase, all of that builds to address the safety consideration. So I would suggest, again, I have coffee mugs at our office for this, but it's to remind them, we have to have the data to make the decision. The agency needs data to make decisions. So having a meeting with a protocol is helpful. It's not going to be the full picture. Anywhere you can apply platform and prior knowledge, I think, is key. Again, if you can start to build that early phase, so let's say you have stability data from a similar product, same manufacturing process, could be the same facility. start to build that in to help support your stability pre-IND and IND. Once you start to have that discussion and you're building that in, it's much easier than trying to surprise a reviewer at commercial. I would say relying solely on DP release assays, not only is it insufficient, I have not seen where any health authority is willing to kind of take that approach. Again, if you have the ability to have platform or prior knowledge, there may be a potential to reduce some of the testing going forward. Small scale models, digital twins, things of that nature, that's also, I think, something in play these days to try to figure out how best you set up these studies, because when you're manufacturing comparability runs, you're not making patient material. The communication with health authority, again, this is a key one. Make sure they understand where you're headed. Make sure you don't surprise them with a PAS submission or a big Type C meeting where you're changing something. If you change strategy after you meet with them, make sure you're kind of circling back to have that discussion. There are many regional pilots at this point. So one, the Advanced Manufacturing Technology Designation. One of the sponsors that's here today was actually the first one in terms of their manufacturing platform. This is a good way to get new manufacturing technology in play. Very early, this would go through the CAT office, which is part of ETT. So by doing that, you can actually get support before you even file your IND. And you can get that technology then reviewed by the agency, talk about the support piece of it, and then it can actually flow into your IND. And then lastly, I would say one of the key items of comparability is that first comparability you do, even though it's early phase and it may have a lower bar, you will be reviewing that in the BLA. They will look at your analytical history, your analytical bridging strategy, and your comparability from phase 1 forward. So don't feel like once you execute that study, it's history. You will be revisiting that in the BLA. So think, I would say, long and hard about the discussions you have with the agency and how you build your strategy going forward. With that, I'll hand it over to Mike. I think we're on time.
Hello, everyone. Thanks, Rupa, for inviting me to this session, and thanks to Karen and Alan for great presentations to contribute to the panel discussion. So before I get into some case examples, and obviously we're not going to be able to get into the weeds in the short period of time, I wanted to break down the comparability approach in what I consider a 3-tier order of stringency, from highest stringency to lowest stringency. And then for comparability acceptance criteria, broken down into 2 tiers, again, from higher stringency to lower stringency. I'm not saying there's not many other examples in between, but just trying to keep it as semi-quantitative and concise as possible. So what's the most stringent? Well, it would be a side-by-side comparison where you're actually manufacturing the pre-change lot and the post-change lots at the same time, as well as testing the material from each of those lots using the same tests at the same time in the same comparability exercise. The next tier, slightly less stringent, would be comparison of candidate CQAs measured on the pre and post-change lots, but you're only manufacturing the new lot and you're using retains from the old lot because you either do not want to or cannot manufacture the old lot anymore. The least stringent of these 3 would be where you're only manufacturing the new lot and either you don't have retains or don't want to use retains from your old lot, and you also either cannot or will not manufacture the old lot again. In any of these cases, which Alan brought up as well, they should always include assays for additional or extended characterization that's not on your lot release panel. For comparability acceptance criteria, again, breaking it down into 2 tiers, the more stringent being based on historical data points for pre-change lot data, where you're actually utilizing statistical tools such as tolerance intervals or standard deviation ranges or equivalence margins or some other appropriate stats approach that would be consulted with a CMC biostats expert. Less stringent than that, of course, would be relying on lot release criteria only for comparability acceptance criteria, and any extended characterization would be based on report results only and part of the discussion in the comparability report. So I broke down case examples into 4. where, again, not being able to get too detailed or in the weeds here, but describing, again, this is just my opinion, other people might have other opinions on what's considered a major manufacturing change, and also what's considered potentially a new product and what might trigger a new IND. Again, knowing that there are guidances like testing multiple versions of a product at the same time to create efficiencies with this, but again, we have a short period of time, so we won't get into too much detail. The pure cell therapy example is the autologous MSC, major CMC change. So it could be same process, but you're manufacturing in a facility in the US and then manufacturing in a facility in Europe. Another example would be the same core process, but moving from a 2D to a 3D or bioreactor manufacturing process. Now, if you wanted to increase the potency of that product by doing some gene edits in that regard, it would almost definitely be considered a new product with a new IND. So what are some of the questions that one can ask, and some of this will come out in the panel? Well, the impact on the timing of the CMC change, right? Does it happen at preclinical stage, clinical, early or late clinical, commercial stage? And then if you go back to what I tried to do is bucket the order of stringencies in A, B, or C, and the comparability acceptance criteria as 1 or 2. Again, I'm not expecting everyone to remember, but just as a way to break it down. As Alan also mentioned, impact of rare disease indication on not doing the classical N of 3 for clinical or commercial stage. What risks have you assessed between the processes and the data sets? As was also mentioned earlier, a risk assessment is a key and initial step in the comparability exercise to ensure that you know which CQAs to focus on at which point in the process and with which stringency. And of course, one should consider combining CMC changes. So if you're making a handful of smaller changes between the first-in-human and the pivotal trial, in a case where there's only 2 trials during clinical development, then you might want to consider combining them into one comparability exercise and the pros and cons of that. Could you combine 2 major CMC changes? Potentially yes, potentially no. Again, as Karen mentioned in her presentation, getting FDA concurrence, thinking about it early, and sometimes you have to approach it from different angles in order to really make sure the approach that you're using is actually going to be successful, not only at that point in time, but once you file your BLA. And then what does comparability success look like? So what's in scope, what's out of scope? For the most part, you have to be meeting your comparability lot release criteria for both the lot release panel and the extended characterization, according to the number of N that you define in your scope, which could be, again, somewhat dependent on the indication. Case example 2, pure gene therapy. So like an AAV. Major CMC change could include a different AAV production platform to enhance yield. But if you went as far as going back to the vector design and changing the sequence of the expressed transgene, that may or may not be considered a new product and potentially a new IND. I won't repeat the same set of questions, but the same set of questions apply to all of these case examples. What about a cell-based gene therapy, like an allogenic CAR-T with gene edits, and what major CMC changes might be associated with that? Well, if you change from a gamma retroviral to a lentiviral platform to actually transduce the CAR transgene, that could be considered a major change. Changing the guide RNA for one of the gene edits to address perhaps off-target concerns that were missed earlier on could be considered a major CMC change. What could potentially trigger it being a new product? Well, if you decided you needed a transgene for a cytokine with the CAR-T, that might very well be considered a new product. Or if you want to do an additional gene edit to make the cells more hypoimmune, that might be considered a new product. And again, the same set of questions apply. The last example is a cell-based combination product. So, for example, autologous epithelial cells on a porcine scaffold, what would be considered a major CMC change? Perhaps changing scaffold from a xenogenetic source to a human source would be considered a major CMC change. But if you now want to move to an allogeneic source and do some gene editing, probably going to be a new product, probably going to be a new IND. Again, the same set of questions apply. So before we get into the panel, just some very high-level thoughts. And before I get there, I'll go back to my first slide because I probably already forgot my own characterization. So again, A, B, and C, A being the most stringent, C being the least stringent for comparability approach, and for comparability acceptance criteria, 1 being more stringent and 2 being less stringent. Sorry. For the preclinical stage major CMC change, you can probably get away with C and 2, which would be the least stringent comparability approach and the least stringent comparability acceptance criteria assessment. For a clinical stage, early clinical likely would not accept that. For clinical stage where you're late clinical will likely require A or B, so higher stringency from a comparability approach perspective, and also higher stringency from a comparability acceptance criteria approach. For a commercial stage, likely require the highest stringency of both for a major CMC change. So what's the message overall? And both speakers previously alluded to this. The later the development stage, the higher the stringency. It's really that simple. For rare diseases, the indications, and this actually applies to any comparability assessment, but especially in the rare disease indications where you're not going to either have or do not want to manufacture the N that would normally be implemented, then getting agreement from the FDA in advance is very important. And again, I should have added that prospective agreement is relevant and important for any comparability exercise, even at the early stages of development. And that's always been a benefit to any program that we've worked on in our consulting firm. With that, we can move on to the panel.
Thank you.
You're welcome. I'm going to move my chair a little bit so I can see everybody. No, that's good, that's good. Thank you. I appreciate the presentations. I love how there was some overlap and then there was also some very distinct points. So in the next 20 plus minutes, we'll go through some questions and then we'll open it up for the audience. So you made the point, but I want to ask the question a little bit different way. So we all made sure to understand that comparability studies are very important from a regulatory perspective, and excessively or poorly designed studies both can pose challenges, right? So under testing will compromise the product, but over testing may actually be considered a new product by the regulatory agency. So is there a sweet spot or is there a Goldilocks where you can guide everybody from your experience to say, you know, what is in scope and what is not in scope? And we can start with you, Mike.
Thank you, Rupa.
Let everybody else weigh in as well. Thank you.
Of course. Can you please present the slide that I'm showing on the monitor? So as was mentioned by Rupa, so what is this sweet spot? So again, if you're in early stage development or preclinical stage, for example, and in my concluding slide I mentioned that would likely be C and 2, which is presented on the slide in front of you, there is really no reason to do more if you can do that amount, right? And especially if you've got FDA's concurrence on that prospectively. In the case of a late stage clinical or either pre or post your pivotal trial, we would definitely not be recommending trying to do anything less than A and 1 without getting FDA prospective concurrence on a comprehensive comparability protocol that contains all your CQAs and the associated comparability acceptance criteria.
Ellen,
You can close the slide now, thank you.
thank you, Mike.
Go ahead.
Yeah, I would suggest when you have those early interactions with the agency, they give you a lot of forward-looking comments, bioactivity, potency. That's going to come into your comparability. So while they may not put a time frame on it, they're pretty much saying the next time we have one of these conversations, you should be ready. And so they do give you a little bit of fair warning, if you will. If you do scientific advice the same way, they're going to tell you what they're happy with, where they're going. I would say when you're doing comparability, one of the big things is you're trying to make sure you're testing that which are your CQAs. You don't need to test things that are not going to actually help you understand whether pre and post change are different. And every time you execute a comparability study, there's, again, all those inherent risks. So you want to make sure the data you deliver from that study is going to support you. But keep in mind, the next time you do a comparability study, your post-change on the first study is your new pre-change. And you generally end up having a higher N on that pre-change than the post-change. So you have to think about that. Your statistician will be your best friend at some point, more than you ever thought, because you're going to have an imbalanced study, and that's the problem. It's not going to be N of 3, N of 3. It's probably going to be 6 or 9 batches versus 2 or 3. So definitely have that conversation.
Great.
Very good point.
A lot of new things already fresh on your mind, but also new things coming up. Would you like to weigh in, Karen?
I think it has to do with logic too. I mean, if you're changing something in your process from, let's say, a manual step to an automatic step, and it really makes a product that is significantly different, then yeah, there's a risk that you will fall into the latter category for a normal kind of change or optimization, no. But I think we all will know when something has significantly changed, and to take a step back as well and say, okay, so if I dosed my pre-change product in humans and got this effect, is it going to be the same in my post-change? Because maybe you've changed an excipient to have it, dissolve somewhere else, whatever. No, you're not going to have the same effect. It's logic to me.
Thank you. So let's talk about starting material. A little bit different ecosystem for autologous, a little bit different ecosystem for allogenic therapies, a little bit different for gene therapies, right? But starting material is very important. And whether it's a patient or whether it's a donor for cell therapies, the variability in that material always poses a challenge. And we hear many times that the most common way to overcome this challenge is to do the split manufacturing or to do paired run studies. And guidelines that you presented are acceptable. Other than that approach, is there any other approach that has been used or can be used in the future? Or, can we ask the regulatory agencies if such thing is not available, what can be done? And we can start with you, Karen.
Yeah, so in the space where we operate, material is that is someone's lifeline.
Yeah.
To do a split batch is taking a lot of the material that someone would need, and the amount of cells and the response is there. So I think there's always a communication to be had and say, no, we can't do this. Out of the best path forward for our patient, I'm sorry, but it's just not feasible. And if so, yeah, there are other aspects, and I think if you have a good justification around, the lack of material, you can look at things like, do you have a well-characterized cohort of clinical subjects? Okay, that married with your statistical programming again, your statistician comes in again. should be able to be sufficient to satisfy some of those needs. But again, it's a conversation to be had, and also for us to sort of put our foot down and say, no, can't do it.
Yeah. Alan, would you like to add anything?
Yeah, I mean, I think at any point, you're going to have a discussion with the agency. You can take any design you think you can support. When you're talking about autologous, so patient-derived starting material, there's always inherent variability. But there's ethical limitations of what material you have, and commercially available material is really not a core material, if you will. So you're going to be using healthy donor to start with. Once you make that choice, everything downstream is impacted. Yeah. Stability, analytics, your reference standard if you have one. So all of those things have to kind of come to play, but that's where you have to have that discussion with the agency. The biggest thing to me is trying to get some variability of that starting material. If you're an allogenic and you make a master cell bank, you probably haven't made a working cell bank. You may get pushed from the agency to think about that, because then at least gives you the variability of the two banks that you could use across comparability. But most companies are not going to make a working cell bank that early, right? Huge cost. Yeah. Testing is exorbitant, but it is one of those things that helps, if you will, from a variability perspective. So your later phase comparabilities, I would imagine for an allogenic, you're going to be looking at master and working cell bank as starting material.
Great. Would you like to add something, Mike?
Sure. Yeah, I agree with what's been said. I think obviously in the case where you can't manufacture a drug product lot because you wouldn't get enough yield if you do split, and obviously that also depends on if you're talking about an allogeneic or an autologous source. Of course, you might be able to do a scaled down model, but representativeness of that scaled down model is going to be very important. And again, as was mentioned, getting that FDA agreement up front, very important. And while it's not necessarily always successful, approaching the comparability run in not only in a GMP manner, but you know, thinking proactively, it might actually end up being able to be used as a treatable for a clinical subject as well, if that helps the process at all, especially if it's something that can be frozen and not a fresh product.
Great. So let's talk about potency assays. Every year we have to talk about potency assays, and hopefully we've learned more than the last year. Or like some of my ARM colleagues will say, we've learned more, but it's more confusing, right? So we have to talk about potency assays and manufacturing change that significantly increases the potency. It could be unintentional, it could be intentional. It's going to raise concerns from a regulatory perspective, right? In cases like that, is it a post-change? Is it a pre-change? How do you manage the critical quality attributes? How do you link those things? What determines all of that? And how do you navigate those things, or how do you set up so that you're not addressing that problem at that point? Maybe we can start with Alan, because you did mention a couple of things in your talk.
So potency, I think, is, as we've seen, we have the updated guidance, which I apologize, I pushed for that for years. But it's now, of course, wanting things earlier in the process. Potency as a whole, I think, is a difficult topic. It is why I would push from a biological characterization perspective. You're really thinking early on about additional assays, surrogate assays, additional models. Because potency as a whole, if you look at, I would say, the last 5 CAR-T approvals, many of them had an analytical or CMC holdup to approval.
Yes.
If you look at the first gene therapy in the US, they were still using an animal model. And then they had to create a release assay during BLA review. So the Spark therapeutic product. That's a very difficult place to be put in. But as you can imagine, the agency didn't learn 2 days before the BLA that that was their potency strategy. So there's also, to me, that communication and coordination across the phases. That is your strategy, but you have to start talking about what's the commercial strategy and make sure that that's clear. Because that is not a place that you can, comparability is one thing, the commercial application is, but it kind of builds on itself. So I would say you need to have the discussion early on. And although potency assurance is kind of laid out as it is in the guidance in the US, even globally, you've got to have a clear biological activity strategy And then your final potency strategy, to me, is kind of the outcome of all of that work. So that's where I think you can have that discussion. And unfortunately, I think biological activity and potency get confused.
Yes.
So to me, potency is a much more late phase discussion.
Yeah. I think that's a good point. Karen, you want to add something?
Yeah, I think my advice is usually to develop that potency as soon as possible, even during your phase 1. I know a lot of people kind of spec and go, okay, well, we don't need it until phase 3. We're fine. Okay, let's start somewhere later on. But it's a long journey to come up with one. It can go all sorts of ways. But also, it makes it really hard for any potential comparability studies if you do not have it. So you're putting yourself in a much more beneficial place by investing in that earlier. And by doing that, you also are going to understand your mechanism of action better and be able to communicate that. So it benefits in many ways.
Great. Would you like to add something?
Sure, yeah. So I think I also agree that earlier the better, because again, you could have blockbuster or breakthrough, first in human data, and then all of a sudden you are approaching late stage without a potency assay or assay candidates. So an early potency assay development matrix approach is always recommended. So in vitro bioactivity assays, surrogate markers of potency that can be correlated together. Therefore, you have more options to choose from down the road, and as CQA being of potency being critical for any comparability exercise, the more you have to work with, the better, and the more data you have to work with, the better. Now, an example of where you increase potency of a product and then ask yourself, well, can this be comparable? If you've already done your first in human clinical trials for dose ranging, and it's going to be pretty hard to rely on that for safety data moving into your pivotal trial if you're going to go with the same dose, because that dose is now more potent. So you might be able to be creative with titrating or diluting your drug product. That's not the road that you really should go down.
Great, thank you. Karen, you showed a slide, and I think both of you alluded to paying attention to analytical assay strategy, which translates into QC assays, and there is no return. If you return back, there are consequences. So what can you talk to in terms of fit-for-purpose assays, and how is the evolution of fit-for-purpose assays changing as you move from the journey from clinical to commercial?
I think it comes down to your product as well, which assays you need. It's very different product to product. Obviously you have to show your basic standard of tests, but otherwise it's really hard to answer sort of generally when a gene therapy product is so very, very different from a small molecule.
Any guidance from both of you?
Go ahead.
Well, I think one of the things to mention is if you're going to be doing one of the less stringent comparability approaches, where you're going to be relying on comparing new test data with historical old lot data, where you're not repeating testing with any revised test, that's a comparability risk in and of itself. So that's where it's important that the appropriate assays are being chosen, and perhaps some pre-work needs to be done prior to the product comparability exercise to confirm that the relevant analytical methods are also appropriate.
Great.
I would just add, I think anywhere you can platform your analytics, the better you are. Each of the modalities has a different analytical package. If you look at gene editing, we went from the first clinical trial to commercial in less than 8 years. you don't have time to be super novel with your analytics. You really need to kind of knock out that what you can, which is kind of low hanging, and then the other stuff is really going to be the biological activity. And you see that now with moving to newer modalities. The expectation that phase 1 IND is raising, the bar has been raised. So I would say anywhere you can platform some of the impurities and other items, that saves you the time to really put your focus on those key assays.
Thank you. One last question before we open it up to the audience. What you said in your talk, Alan, struck me. What can you tattoo on your forehead?
I'm telling you.
So I'm going to ask all three of you, starting with you, Karen. What can your coffee mug say? Or maybe nobody wants to tattoo on their forehead, but something. What is it that you always keep in mind, or what is always on top of your mind when it comes to challenges associated with comparability studies, or something that you've done well? Just like a phrase that people can take home with them.
Plan ahead.
I like that.
Years, and outline it, you know, and make sure that you do it in a smart, logical manner. What do you do first? What do you do later? What influences what? Put it on there. Make everybody aware that this is coming.
Great. Alan?
So I'll change my answer for my slides, but I would tell you to think holistically. Those of you who develop these products and work on them, you know them better than anyone, but you know them better than the health authorities you deal with. So you've got to step back, give them the proper story, give them the right background. And honestly, sometimes stepping back, you'll see kind of the bigger picture approach as well. So I would tell you, it's hard to do. I know everyone's got a timeline, but once in a while, we pull our teams aside. I'm like, I want someone who doesn't work on that program to look at the submission. And it's a whole different view, because you just want someone to look, is it readable? Do you feel like you started in chapter 2 of the story? Those kind of things can really be beneficial.
Great. Mike?
Agreed. FDA prospective concurrence on a t-shirt, maybe not a tattoo.
FDA will be happy. Well, we really appreciate your presentations and a very insightful discussion, and like for you to join me in giving our panelists a big hand. We have a few minutes for questions, and our lovely Natalie is going to pass on the microphone. Thank you, Natalie.
And if anyone wants to debate or pressure test the ABCs and our 1s and 2s,
Mike is
please feel
available
free.
after this for a debate.
Okay. Hello, thank you very much for this talk. You've mentioned before have you ever considered the bias that might be in your donor starting material between like gender and race? I know with rare diseases you tend to have a specific type of patient, but when you have those outliers, do you try to incorporate that into your data? And when you do get that data, how do you proceed after that?
So I guess I can start.
Sure.
It's a great question, and it's actually one that we look at. It depends on the disease. So for certain diseases, they're going to be in certain populations more, but it's one of the biggest risks that most developers face, which is you have a certain population in a certain region of a country, and then you get all of these commercial products that have different variability. So countries like Japan, you have to do a clinical study there for that reason. They made it a part of it. The US, they do have some clinical trial diversification to make sure that we try to broaden that out. What you're seeing in oncology, though, there's kind of limited footprint. We've kind of saturated, so I think that's what you're seeing here today is, can we get this more into rural and other areas? That makes it more equitable in terms of the patients we'd have in those pivotal studies. But it's a key item. If I don't see it in the manufacturing, when I set commercial specs, I then get surprised in the commercial space. So it, to me, is a highly relevant thing for the development space.
Yeah, I agree. It's a great question, and some of it, unfortunately, is the limitations of the starting material sources. So if you're a small company and you need to source your biological starting material, and you don't have in-house apheresis capacities or something similar, oftentimes there's maybe limited selection based on where your geography is of what type of donor you're going to get, depending on what your starting material is going to be. So it's definitely worth being proactive on, especially as you go from early to late stage clinical development. So great point.
Hey, this is Bing Wu from Ultragenics. So my question is about the gene therapy comparability. So when you have very limited number of lots, what's your approach? Because the background is you're trying to demonstrate your comparability pre and post change statistically, not just to compare one lot to another lot. So there's some expectation from agency that you statistically demonstrate your comparability. So the question is, we don't have many lots. How do you conquer this?
It's definitely a question no one wants to address. But I think important to note is the rare disease space, right? This does come up a lot. It does come up a lot for AAV. And there are consortiums like the BGTC that are also trying to address some of these issues with AAV rare diseases as a playbook, so to speak, for what can be done by others. But I think getting FDA concurrence, again, is the most important thing. So yes, from a practical perspective, and when I said in my presentation, either cannot or will not manufacture, that's a little bit different than, cannot and will not are a bit different. But at the same time, in a limited resource environment and limited pre-change runs, the FDA will be practical. It's just coming up with a storyboard that makes sense. And as far as the statistical approach, I'm forgetting the word, but there is a word for when you don't have, the ability to do tolerance intervals or SD ranges. There is really just a sort of a head-to-head comparison. There's a word for it that's escaping me. I don't know if anyone else remembers.
Skewed statistics? Oh.
No, maybe somebody in the audience knows which word I'm looking for.
That a quiz, Mike. Yeah.
If you're meeting with the agency, I would suggest if you have like an engineering run where you can show similarity to your clinical process from that, even non-clinical data, you have to try to bolster it the best you can, but you've got to show that material is, you don't want to put yourself in a deficit where you use a material that's already not similar to your clinical batch, but you may be able to build that story, especially if this is, the same AAV and the same manufacturing process. But to Mike's point, the Bespoke Gene Therapy Consortium is a great place. They do have an initial IND handbook out now, so, and I don't personally support it, so I'm not promoting it, but it's a really nice way to kind of align approaches, because if not, I think Peter Marks used to say this when he was at FDA, was everyone has a different approach. We need some consolidation, because every time they got, a new product is a different way. So anything you can learn from that, I think would help build your strategy.
Great. We have time for one quick question. 2 minutes and 30 seconds left.
No pressure testing on the ABCs and the ones and twos?
Uh-oh.
I could ask, but yeah. No, but I guess maybe a question is that, as you mentioned the assay methods, and it's kind of locking them in early. But as you go through from clinical to commercial, the assay methods they really can change as you get to know things about the actual tech can also change from qPCR to ddPCR. So how do you look to approach that when you say you want to change the assay method? Do you look to do it pre-commercial or post? And then what sort of comparability we would look at as well?
I mean, I'd highly suggest you don't change methods post-approval, but I think before pivotal is definitely at the time, but you can do it during the process. You're going to have to bridge the methods, so you'll have to kind of test old method, new method, pre and post, so you're going to need a material. But it's, especially if you're seeing that you have variability in the current method and you're approving on the technology, the agencies are generally on board, because that's going to be a release method with specifications. So, but it could require a lot of material, so you'll have to kind of prep in advance, and you may need some stability testing as well.
Mm-hmm.
Great. Thank you so much.
Thank you.
Wonderful.
Thank you.
Thank you. There's one after this.